Welcome to the deep dive. Today, we're really getting into the weeds on a particularly challenging cancer, pancreatic ductal adenocarcinoma, PDAC.
It's notoriously difficult, yeah.
And we're not just looking at the tumor cells themselves, we're focusing on the stuff around them, the support system, the fortress they build.
Exactly. That fortress, the tumor microenvironment, or TME, is often the real barrier to treatment. It's dense, it's protective.
And our source material today, a key study, really digs into the cells that build this fortress, these cancer-associated fibroblasts, or CAFs.
That's right. These CAFs are the main architects of that physical wall that stops immune cells, stops drugs.
Yeah.
It's a huge problem in PDAC.
So the big question this study tackles is, what if we could mess with the builders? Take away a key tool, or maybe their reason for building?
Precisely. The research suggests there's one specific process, macropinocytosis, that acts like a survival shield for these barrier-building CAFs. It keeps them in that builder role.
An identity shield, they called it.
Right.
So if you take away the shield,
You force an identity crisis. You inhibit macropinocytosis, and these CAFs dramatically switch jobs. They start building the wall and become, well, different, more inflammatory.
turning the fortress into more of a battlefield.
That's a great way to put it. It potentially makes the whole tumor much more open to attack by therapies. It's all about metabolic reprogramming.
Okay, let's unpack that. Starting with the environment itself. Why is it so tough in there? Why do these CAFs need this intense survival shield, this macropinocytosis thing?
Well, the PDA-TME is just hostile. Think resource scarce. The researchers actually measured the fluid around the tumor cells in mice.
And what did they find?
Really low levels of nutrients, critically low glutamine. Compared to normal blood plasma, it's practically a famine zone for glutamine, which is a vital amino acid cells need.
So the cells are under serious metabolic stress from the get-go, starving, essentially.
Exactly, constant stress. And they need a way to cope, to find alternative fuel sources.
Is that where macropinocytosis comes in, like an emergency ration system?
That's the perfect analogy. It is the emergency kit. Macropinocytosis allows the cell to just gulp, take big, nonspecific drinks of the fluid outside the cell.
Fluid that contains proteins, like albumin.
Right. It pulls in whatever is out there, including proteins like serum albumin. Then it breaks those proteins down internally into amino acids. It's a way to scavenge for building blocks when the normal supply lines, like glutamine, are cut off.
A desperate measure to stay functional.
Absolutely. Both the CAFs and the tumor cells themselves use it to survive this nutrient-poor environment.
Okay, scavenging makes sense. But you mentioned CAFs aren't all the same. They have different roles, right? Heterogeneity.
Yes, exactly. CA heterogeneity is key. We mainly talk about two subtypes here. First, you have the myofibroblasts, or myCAFs.
Myo, like muscle.
Sort of. They express proteins like alpha-smooth muscle actin, alpha-SMA. Think of them as the construction crew. Their main job is producing and depositing the dense extracellular matrix, the ECM. Lots of collagen.
So they lay down the concrete and steel beams of the fortress wall. Collagen types like COL1A1, COL1A2.
Precisely those. That dense collagen matrix is the physical barrier that literally blocks T cells from getting in and stops drugs from reaching the tumor cells effectively.
The physical defenders. Got it. And the other type.
The other main type are the inflammatory CAFs, or iCAFs. Their role is more about communication. They secrete signaling molecules, cytokines, chemokines, things like interleukin-6, IL-6, CCL2, CXCL1.
Signals that usually call in immune cells, right? Although maybe not always helpful ones in cancer.
It's complex, but yes, they send out inflammatory signals.
Okay.
So you have builders, myCAFs, and signalers, iCAFs. The big mystery was, does this metabolic stress, this need to scavenge, influence which role a CAF takes on? Can you force a builder to become a signaler by messing with its food supply?
So connecting the metabolism to the cell's identity or function, how did they test that? Did they try to block the scavenging in the lab?
They did. They set up an experiment in vitro, growing CAFs in a dish. They mimicked the tumor environment with low glutamine.
Okay, putting them under that starvation stress.
And then they added a drug called EIPA. EIPA specifically inhibits macropinocytosis.
So step 1, starve them of glutamine. Step 2, block their emergency scavenging route with EIPA, a real double whammy.
Exactly, a metabolic pressure cooker. And the first thing they checked was, did this actually increase the stress?
Yeah, did it?
Oh yeah. They saw levels of a protein called SESN2 shoot up. SESN2 is like a cellular alarm bell for amino acid deprivation. So blocking macropinocytosis definitely made the starvation worse. It proved macropinocytosis was the lifeline.
Okay, proof of concept. It's a vital survival tool. What happened to the CAF identity then? Did the builders stop building?
That was the crucial result. Under low glutamine plus EIPA, the markers for myCAFs, the builder genes like ACT2 and teglin, dropped significantly.
They stopped making the construction materials.
And simultaneously, the markers for iCAFs, the inflammatory signaling genes like IL-6, CSGL2, IL-1B, went way up. They switched roles.
Wow. So they reprogrammed from structural support to inflammatory signaling. But hang on, how do we know the myCAFs didn't just die off, leaving only the iCAFs behind? Was it a real switch?
Excellent question. They checked that very carefully. They looked at cell viability and proliferation markers, like Ki-67, and they found no significant difference in cell death or growth rates between the subtypes under these conditions. It wasn't just selection, it was genuine reprogramming. The myCAFs were actively changing their identity into iCAFs.
That's a critical distinction, true plasticity.
And they even showed it was reversible.
Yeah.
If they took away the EIPA and gave the cells plenty of glutamine again, the cells would shift back towards the myCAF state over time.
So the myCAF identity isn't fixed. It's actively maintained by their ability to use macropinocytosis in that stressful environment. Take that away and they're forced to adapt.
Precisely. It highlights how dynamic these cell states can be.
Okay, so we know what happens, they switch identity. But how? What's the molecular switch inside the cell that gets flipped when macropinocytosis is blocked under starvation?
They track that down too. The key signaling pathway involved is the MEK-ERK pathway.
Ah, a common pathway involved in cell growth and stress responses.
Exactly. When they subjected the CAFs, both mouse and human CAFs, to that double whammy of low glutamine and EIPA, they saw a significant increase in the activated form of ERK phosphor K.
So the pathway lights up under this specific stress. How did they confirm it was required for the identity switch?
They used inhibitors, drugs like tramitinib, which blocks MEK, or another one called SCH-772984, which blocks ERK directly.
Standard tools for blocking that pathway.
Right. And when they added these MEK-ERK inhibitors, along with the low glutamine and EIPA, the increase in those inflammatory iCAF genes, it was suppressed. It didn't happen.
So blocking MEK-ERK prevented the switch. That confirms it's the essential downstream pathway. Starvation plus no scavenging triggers a MEK-ERK, which then drives the reprogramming to an iCAF state.
That's the mechanism they uncovered. And importantly, they found this was happening independently of a signal called interleukin-1 alpha, IL-1 alpha.
Why is that important? Wasn't IL-1 alpha thought to be a major driver of iCAFs, usually coming from the tumor cells?
Yes, much of the previous work suggested iCAFs were induced by external signals, like IL-1 alpha, released by other cells in the TME, what we call a paracrine mechanism.
So the CAFs were just following orders from the tumor.
That was one model. But this study shows an intrinsic mechanism. The CK itself senses the metabolic stress, the lack of glutamine, combined with the inability to scavenge via macropinocytosis, and it triggers its own reprogramming via MEK without needing that external IL-1 alpha signal.
That's a big deal therapeutically, right? It means you might be able to trigger the switch just by targeting the CAF metabolism directly.
Exactly. You don't necessarily need to block signals coming from the tumor cells to induce this specific change. You just target the CAF's internal survival strategy.
Okay, this is compelling in the dish, but the real test is in a living organism, right? Did these changes translate to actual effects on the tumor and its environment in mouse models?
They moved to orthotopic PDA models, implanting tumors in the pancreas of mice, which mimics the human disease setting more closely. And they treated these mice with EIPA.
First off, did it affect tumor growth overall?
Yes, EIPA treatment did reduce overall tumor growth, which is good. But more importantly, for this study, they looked inside the tumors at the CAFs.
And did they see the identity switch happening in vivo?
They did. Consistent with the in vitro data, EIPA treatment led to a decrease in the myCAF population and a significant increase, about 2.3-fold, in the iCAF population within the tumors.
The switch holds up in a real tumor environment. Did they look deeper, maybe with single-cell analysis?
Yes, they used single-cell RNA sequencing, saRNA-seq, which gives you a really high-resolution picture of individual cells. This revealed something fascinating.
What was that?
It wasn't just a simple flip from myCAF to iCAF. The data suggested a more gradual transition, showing cells in an intermediate state, sort of a hybrid myCAF-iCAF identity.
Ah, so a transitional phase, like the cells are halfway through the reprogramming.
Exactly. And using computational tools like pseudochem analysis, they could map out this differentiation trajectory. myCAF flowing towards this intermediate state, and then onto the iCAF state. EIPA treatment actively pushed cells along this path.
That level of detail is amazing. So the decrease in myCAFs, did that lead to actual physical changes in the tumor structure? Did the fortress walls start to crumble?
That was the critical question, and the answer was yes. They looked at collagen deposition using staining techniques like trichrome staining.
Which stains collagen blue, right?
Right. And in the EIPA-treated tumors, there was significantly less blue staining, less collagen. The dense matrix was genuinely reduced.
So literally dissolving the physical barrier. What about blood vessels? You'd expect them to be compressed in that dense environment.
They were. But with EIPA treatment reducing the myCAFs and collagen, the physical pressure eased up. They stained for CD31, a marker for blood vessel endothelial cells.
Yeah.
And they saw significantly expanded tumor vasculature, more open, functional blood vessels within the tumor.
That makes perfect sense. Less dense tissue, less squeezing, the vessels can open up. That has huge implications for therapy, doesn't it? Better access.
Absolutely. Two major avenues opened up: better immune cell access and better drug delivery. Let's take immunity first.
Okay, the T cells. If the collagen barrier is reduced, they should be able to get in easier, right?
That's exactly what they found. EIPA treatment led to a marked increase in the infiltration of both CD4 plus helper T cells and CD8 plus killer T cells into the tumor. About a threefold increase in total T cells.
Wow, 3 times as many soldiers getting past the walls. Did they confirm the reprogrammed CAFs were actually helping attract them?
They did a neat experiment. They took the culture medium, the fluid, from CAFs that had been treated with EIPA in the dish. This conditioned media contained the factors secreted by those reprogrammed iCAFs.
And they put T cells in it.
They used it in a migration assay. And yes, T cells migrated much more readily towards the conditioned media from EIPA-treated CAFs compared to controls. It shows the iCAFs were releasing chemoattractants, signals actively calling the T cells in.
Okay, so you've got fewer physical barriers and active recruitment signals. That sounds like a perfect scenario to combine with immunotherapy like checkpoint inhibitors.
Exactly the rationale. They combined EIPA treatment with an anti-PD-1 antibody, a standard immune checkpoint blockade, ICB.
And the results. Did it boost the anti-tumor effect?
It did, particularly concerning metastasis. The combination of EIPA and anti-PD-1 significantly reduced the spread of the cancer, fewer large metastases in the intestine, and fewer tiny micrometastases in the lungs.
That's a major win, as metastasis is what often makes PDCs so deadly. Did it improve survival?
It led to a modest improvement in overall survival in the mouse models. Interestingly, the T cells that did infiltrate still showed signs of exhaustion, which is a common challenge in PDA. But suppressing metastasis is still a very significant outcome.
Definitely. Okay, so that's the immune angle. What about the other implication of the remodeled TME, the expanded blood vessels? Did that help chemotherapy get in?
That was the hypothesis. More open vessels should mean better delivery of chemotherapy drugs to the tumor cells.
How did they test that?
They used doxorubicin, or DOX, a chemo drug that happens to be fluorescent, making it easier to track. They gave mice EIPA first to remodel the TME.
To open up the highways.
Exactly. And then they administered DOX. They found significantly higher concentrations of DOX accumulating inside the EIPA pre-treated tumors compared to control tumors.
So the drug delivery was genuinely enhanced? More chemo getting to where it needs to go?
Precisely. The remodeling worked as predicted.
So the ultimate test, combine EIPA pretreatment with a standard PDAC chemotherapy, like gemcitabine.
They did that combination. EIPA pretreatment to remodel, followed by gemcitabine, which is a first-line drug for PDAC.
And the outcome, synergy.
Clear synergy.
Yeah.
The combination was significantly more effective at suppressing primary tumor growth and reducing lung micrometastases than either treatment alone. They calculated the combination index, or CI.
Which tells you if the effect is additive, synergistic, or antagonistic.
Right. And the CI was less than 1, which is the mathematical definition of synergy. The combined effect was truly greater than the sum of its parts. Remodeling the TME with EIPA made gemcitabine work substantially better.
That's incredibly promising. Okay, let's try to synthesize this. It seems like macropinocytosis isn't just a lifeline for CAFs, it's what keeps them locked in that builder mode, maintaining the tumor's defenses.
That's the core takeaway. It acts as this identity shield for the myCAF estate.
And if you inhibit it using EIPA or potentially other methods, you break that shield. You force the SUCAFs through that MEC pathway to reprogram into iCAFs,
Right. They stop building the dense wall and start sending out inflammatory signals.
which has multiple benefits. The physical barrier, collagen, decreases, blood vessels expand, and T cells are recruited in. It fundamentally reshapes the TME.
Making it much less protective for the tumor and much more accessible for both immune cells and chemotherapy drugs.
So the big picture here is that targeting the stroma, specifically CF metabolism, is a critical strategy for PDA. It's not just about killing tumor cells directly.
Absolutely. This strongly supports a dual approach. Target the tumor and target its environment. Inhibiting macropinocytosis looks like a very promising way to tackle the environment to make existing therapies far more effective.
It really shifts the paradigm. Now, thinking ahead, you mentioned the reprogramming was reversible and the single-cell data showed that intermediate state. That raises a question for you, the listener, to ponder.
Right. We know these cells are plastic, they can change back and forth, and they go through steps. So the question becomes one of timing and optimization.
If you inhibit macropinocytosis, how long do you need to do it for? Is there an optimal window to get the maximum TME remodeling, the barrier breakdown, the vessel expansion?
Could inhibiting it for too long allow the scavs to adapt somehow, find another way, or maybe even revolt spontaneously? Is hitting them briefly enough to initiate the changes needed for combo therapy?
Finding that therapeutic sweet spot, the right duration, the right timing, in combination with chemo or immunotherapy, that seems like the next critical step to figure out for translating this into the clinic. Something to keep an eye on.